Welcome to Biology Minds. Today we're going to talk about DNA pertaining to forensic science. So whenever we think about forensic science, we're always talking about fingerprints and DNA. So the big thing now, as much as all the old types of evidence are very important, DNA is the new thing because it is the new smoking gun. It's the way that we can pinpoint who was at the scene, what happened, and we can help to reconstruct our crime scene. So as important as it is to still collect all the other evidence and to build our case around the other evidence, we really are always looking for DNA now because it is so helpful in getting our perpetrators to justice. So humans have 46 chromosomes. Just as a general review, remember you're getting 23 of them from your mother and 23 from your father, which accounts for about 30,000 different genes. So when we talk about DNA, we talk about having these nucleotides, you have these nucleic acids, and there's going to be about 3 billion base pairs, okay? But each base pair is not going to code for a specific characteristic. It really comes down to a length of base pairs, which we can call a gene. So when we have these different lengths, they're going to code for different traits, different characteristics, and they're out of those 3 billion base pairs, it creates about 30,000 genes. So you have 30,000 genes, I have 30,000 genes, but we're going to have different genes, and that's what's going to make me different from you. So genes are the fundamental unit of heredity. They're going to instruct the body cells to make proteins that determine traits such as hair color, personality, even susceptibility to diseases. So we always think about DNA as being our nitrogenous bases, our A's, our T's, our C's, our G's, but they are going to make our mRNA, which is going to make our U's, A's, G's, and C's, and then that's going to be, this can be used, this is our mRNA, this can be used as the instructions to make our amino acids, and amino acids are the building blocks of proteins. So you might not think of it this way, but DNA is going to create the protein. So that is how, it's not that the DNA is necessarily saying, hey, you're going to have this color hair, but it's going to help build the proteins for your hair, and that's how we see the correlation between DNA and what characteristics or what traits you may have. So DNA is deoxyribonucleic acid. All right, it has a deoxyribose sugar in it that makes up this nucleic acid. It also has a phosphate group attached to it. And then it has these nitrogenous bases. It has adenine, thymine, cytosine, and guanine. And remember, if you remember from 9th grade biology, living environment, A is always going to go with T in DNA, T is always going to go with A, C is going to go with G, and G is going to go with C. So if we have one side of the DNA, then we know what the other side is going to be automatically. Remember, it is a twisted ladder. When we look at it, it's that double helix. When we look at it, sometimes we look at it untwisted, but in reality, it is always this twisted double helix, this twisted ladder that is made up of your sugar backbone with phosphate groups attached to it, and in the middle, connecting the two sugars, is going to be these nitrogenous bases that are A's or T's or G's or C's. So any change in that we're going to call a mutation. That's going to be a change, or we can call it an error in DNA. And this just shows you how we are going to build our proteins. So in our nucleus, we are going to have our DNA. This is our DNA. And whenever you think about DNA, DNA is going to stay in the nucleus, okay? If you want to think about it as the most important piece of paper that your cell has. If you have, say, the Bible or something that's very important to you, it's the Holy Grail, you're not going to let someone borrow your Bibles. You only have one left in the world that it's very important to you. You're not going to let someone borrow it, but you might let someone make a copy of it and then take the copies with them. All right. So what the nucleus does is it says this DNA is too important. You're not going to take the DNA out of the nucleus and do whatever you want with it and then maybe return it later. No, we're going to keep the DNA here, but we're going to make instructions to send to the ribosome so that they can build these amino acids, they can build these proteins. So what we do is we make mRNA. Okay, so we're going to make our mRNA, and the mRNA instructions, okay, this is going to be sent to our ribosomes, and then the ribosomes are going to read these instructions, and based on the instructions, based on which nucleic acids there are there, or I'm sorry, which nitrogenous bases are there, it's going to know, hey, we have to add this amino acid to this amino acid, and then this amino acid is attached to that one, and that's how we build our proteins. Okay, this is showing you, hey, we have these instructions, and we are building our proteins. So when we talk about forensic science, we have to talk about, okay, how are we using this DNA to help us in investigations? What can we do with this DNA in order to process it from the scene in the laboratory and use it for our investigation? And a big thing, a big piece of technology that came along was this polymerase chain reaction, which we abbreviate to PCR. So PCR is a big thing because it is multiplying DNA. So if you have a very small sample of DNA, once upon a time, you had a small sample of DNA, you used it for testing. Once you used it up, you had no more. But now we use this PCR, and we're able to make copies. So we're using DNA polymerase, okay, which is an enzyme responsible for unwinding and proofreading DNA, and it's used by us to make a copy of DNA strands located outside of the living cell. So as much as it is used inside the cell by itself to make those copies, we are using it to make copies ourselves. So small amounts of DNA can now be multiplied using PCR, and when we put it into a thermal cycler, and now suddenly sample size used to matter. Now there's no longer a limitation. You have as much DNA sample as you need. All right, we're making copies of that DNA, and as we have more and more copies of the DNA, now we can test it as many times as we want. We can do different tests to it, and we don't have to worry about, well, once it's done, it's done. Once we ran out of it, we ran out of it. So we now, now that we have PCR, we're going to use it for any DNA evidence that we have, so that we have plenty of sample to use. And recombinant DNA is going to be using restriction enzymes to insert or remove genes in a DNA strand. So we talk about recombinant DNA whenever we're talking about gene editing, changing genes. So a famous one is in bacteria. Okay, in bacteria, we've actually been able to take genes out of bacteria and fit in human genes. So now they have the code for how to make insulin, and that's how we actually make insulin for diabetic patients, humans that need that insulin. They need to inject insulin. So previous to that, they had to use insulin from pigs or some other animal, and they had to harvest that from them and then give it to people. But then you run the risk of maybe an allergic reaction, some adverse effects. Now we're taking human insulin that is made by a bacteria, but it's exactly the same because we're using that same exact genetic code to make those amino acids, to make that same exact protein as the human insulin. So we're making human insulin using bacteria, and now we are able to create that insulin for diabetic patients and give it to them just by changing the genome of the bacteria. This just gives you a diagram that shows you that, hey, we have a human cell and we have a bacterial cell, and we are going to cut out a gene from the human DNA, and then we're going to make room for it. We're going to cut out a little piece of the genome of the bacteria, and then we're able to insert this DNA into this space, and now we have this new bacterial DNA, and now suddenly this bacteria is able to make its own insulin, or in this case they're talking about HGH. It's able to make it because we inserted the gene that gives the instructions to the ribosomes of how to make those proteins, how to make those put those amino acids together to make those proteins. So DNA typing, the old way of doing it, all right, what really in the 90s when they were first starting out, the big thing was RFLPs, these restriction fragment length polymorphisms. More than 30% of DNA is made up of these tandem repeats. Okay, tandem repeats, all right, they're bases that are repeated numerous times, and they're not meant for producing amino acids or proteins. They're called non-coding segments. All right, and the non-coding segments, they don't really do anything for us per se. They're kind of placeholders for the next gene that is going to code for something, but we're able to use them to compare one DNA to another person's DNA. So we use these tandem repeats, and everyone has these different tandem repeats, but if we look at the different lengths of them, we can compare different DNA samples. So all people have the same type of tandem repeats, but there are a lot of variations in the number or the amount of repeats. Okay, so everyone has these same tandem repeats. Okay, for example, if I had, say I had I'll make up a name, I had a TAS4 repeat. Okay, and in me it repeats 78 times, but in someone else it repeats 63 times. That would be one way to compare them. You say, oh, this is not the same DNA because I looked at this DNA sample and the TAS4 was a 78, and then when I looked at this sample, the TAS4 repeated 63 times. So it's the same section of the DNA, the same segment of the DNA, but there's a different amount of nucleic acids, a different amount of letters that we're going to see there. So practitioners use these restriction enzymes, which are like the genetic scissors. That's how we're going to be able to cut out a gene from the bacteria and insert one of our genes. We use these genetic scissors. There are no little microscopic, tiny, they'd have to be more than microscopic, tiny scissors that we can cut up DNA with. So we actually use these enzymes, which are going to, they're called restriction enzymes, they're going to break up the DNA for us, and that's how we're going to be able to cut up the DNA into specific segments. So with RFLPs, we cut up the DNA, and then we're able to look at how do the strands compare in length, all right, using thousands of bases that are thousands of bases long. So this gives you kind of an overview of what you're doing. So you're collecting the evidence from the crime scene, and then you're going to isolate your DNA from the evidence sample. And once you've done that, you're going to cut the DNA into fragments using these restriction enzymes. For every person, the size of the cut fragments are going to be unique, so that is important. So although we all are going to have these different segments, they're going to be different lengths. So then when we cut them up, we can compare one sample's lengths to another sample's length. So you separate the negatively charged DNA fragments in a gel by passing an electric current through it. Okay, we're going to see that we have these negatively charged DNA particles, and they're going to move towards the positive side of the gel. You're going to transfer the DNA fragments from the gel to a sheet of membrane, and then you're going to probe the membrane with DNA fragments that complement the DNA sequence of the fragments of interest, and you're going to compare the fragment profile of the evidence DNA with those of the suspects, the detective, the victim, anything that you're going to think is relevant. Any suspects, anyone that might have been at the scene, you're going to compare them. So here you'll see that they give you an example. So you have your evidence DNA, and we have one RFLP, and we have another one right here. And these are the two lengths based on how far they went. When we look at a gel electrophoresis gel, we're going to see that the larger they are, the slower they move. So as we're moving this way, anything that's very large is going to stop sooner. So this was large. And then anything that's very small is going to make it towards the end. So when we're looking at the different suspects, all right, we're going to compare. All right, this was our evidence DNA, and we're going to compare. Well, this suspect, they had a very long one, and then they had a much shorter one. This victim had this very long one, and then somewhere in between. The officer had a medium one, and then a very short one. But if we look at suspect 2, they had the exact same lengths as the evidence that we found at the scene. So suspect 2 is our person, or we could say at least it is the same DNA as this evidence DNA. So then we're going to reprobe the membrane up to 10 more times to identify different fragments. If the profiles from the evidence DNA and the suspect match multiple times, then it's very likely that the evidence DNA came from the suspect. So when we're looking at this, all right, we're going to want to check our work. We're going to want to do it more than once because it's very important in order to, when you go to court, to prove that, hey, there was no manipulation, there was no issue with our technology, with our equipment, and that we know for a fact that that evidence matches the suspect's DNA. So gel electrophoresis is this process used to separate the fragments of the RFLPs into different lengths. By placing different samples into different wells, you're going to have different lanes. So if you go back, you see that this was one lane right here, and then this was another lane, and this was another lane, and it's all by putting these different DNA, okay, in these different wells. So there would be these little areas where we insert our DNA. All right, we're going to have our DNA all mixed up. They're going to be broken up into different fragments, but they're going to be all together in one sample, and we're going to see that they're going to separate as they move down the gel. So by placing different samples into the different wells, then you're going to see how these ones separate, how these ones separate, how these ones separate as they move down. So to identify the specific sequence of each fragment, a nylon sheet is treated with this radioactively labeled probes containing base sequence to complement a sequence that is trying to be identified, which is called hybridization. So we need a way to pinpoint what is this sequence, and we use it using these radioactively labeled probes. The probes act as a marker since it is labeled, and when a probe complements or joins a sequence, since we know the sequence of the probe, then we know the other side. So if we have a probe that is going to be A, T, C, and it's going to link up with, say, 1, 2, 3. Well, we know that, hey, 1 has to be a T because A always goes with T, and 2 has to be an A because T always goes with A, and then 3, our third one, has to be a G. So we know just by them pairing up that, hey, this, our probe was, our labeled probe was ATC, so that means that our DNA that it linked up with must be TAG, and that's how we're able to figure out what exactly the sequence is that we're looking at. Because when you look at the gels, you just see these bands of DNA. All right, it's not gonna jump out to you and read the letters to you, so you have to use these probes in order to figure out what the letters are. So once the nylon sheet is exposed to X-rays for several days, it shows the results from the DNA probe, and by comparing the two separate samples, we can see if there is a match. By using several probes, the degree of discrimination or even individualization, if you want to talk about how DNA is going to pinpoint one specific person's DNA, it greatly increases. So the new standard is using PCR, and once you make those copies, you're going to then do SDR analysis. So as much as RFLPs were a major breakthrough with using gel electrophoresis, now we don't use RFLPs as much as we use STRs. So STRs are the new standard. And in PCR, DNA is going to be heated to 74 degrees Celsius to separate the two strands, and then a primer, which is a short strand of DNA made by scientists to target a region of DNA for replication, is going to be added to hybridize or to combine with the strands, and this lowers the test tube temperature. Then the DNA polymerase and the nitrogenous bases or the nucleotides are added. The test tube is heated to 72 degrees Celsius, and the DNA polymerase rebuilds each strand. So one DNA is then becomes 2, and by completing the cycle 30 times, over 1 million copies are available, because 1 becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16, 16 becomes 32, and then after 30 times of doubling everything up, you're going to end up with 1 million copies. So PCR cannot be used with RFLPs because the strands are too short, so we use STR instead. All right, STR is the better way of going about it. So the new standard is using these short tandem repeats, this STR analysis. So STRs are locations known as loci, on the chromosome that contain short sequences that repeat themselves within the DNA. So they are made of less than 400 bases, so significantly shorter than the RFLP. So where you see that the RFLPs were these long strands of DNA, so DNA, we said it has 30,000 genes, and we can cut it up into these lengths of 100, 200, 300, 400 bases to make our RFLPs. Well, now we're looking at STRs, which are a lot shorter than that. So whenever we look at someone's STRs are the same idea where it's going to be the same location in the DNA. Everyone's going to have those locations, but there's going to be different amounts of repeats. So this guy, for STR1, he's got 10 repeats, and for STR2, he has 12 repeats. For this young lady, she has 14 for STR1 and 7 for STR2. So we're looking at the same areas of the DNA on each person, but we see that there's different amounts of repeats, and then he's going to have 5 and he's going to have 10. So just by looking at these three people, we see that, hey, they have very different STRs between each other just by looking at two STRs, two little segments of DNA. So the pros of STR is that it's less susceptible to degradation, can be recovered from bodies that have extremely decomposed, and can be duplicated using PCR. So as much as we like to use PCR in general, we need PCR, or PCR is even better for these short tandem repeats. Only 1/1000th of a gram sample is needed, so you need next to nothing, next to no DNA in order to use STR analysis. So STR is used with gel electrophoresis again, just like RFLPs are, to see variations from one sample to the next. Current technology allows for multiplexing, which is simultaneously detecting more than one DNA marker in a single analysis, so we can do several tests. By combining results, we can decrease the chances of DNA belonging to someone else. It becomes individual evidence. No longer is it class evidence. So remember the product rule. The big thing with STRs is we're using this product rule where we're multiplying the chance of this by the chance of this by the chance of this, and then it's making the chances much and much smaller. So if we get a readout like this, okay, this will show you that, all right, for this specific STR, it's named D5S818, and this individual has a 10 repeat and a 12 repeat. And then we look at this one, it has an 11 and a 12, and this one has a 10 and 11, and so on and so forth. And then we see if we were able to look at just 13 STRs, 13 little segments of the DNA, 13 sequences, excuse me, we're able to yield individual evidence. So in our CODIS, which is our database for DNA, we are going to document or have data on anyone that's ever been put into the system, and all we're going to have is we're not going to have the entire printout or readout of their entire DNA. It's going to be just these 13 STRs, and that's all it takes, because all 13 yield a 1 in 575 to 900 trillion chance of matching another individual. There's only about 110 billion Homo sapiens that have ever lived. Okay, there's about 7 billion people alive right now. Okay, if there's over 7 billion, okay, we're talking about not 900 billion, we're talking about 900 trillion. So that number is so much larger, it would be impossible to see that someone else has those same exact 13 STRs. So even if we look at only 3 STRs, 3 little segments, we're going to see it's a 1 in 500 chance. Once we get up to 6, it's a 1 in 2 million chance. And then once you get to 9 STRs, it's a 1 in 1 billion chance. So the chances just right there by looking at 9 STRs is very unlikely, because maybe there's only 7 people in the world that are going to have those same exact 9 STRs. And then if you have a specific suspect, well, what's the chances of that suspect being there and 2 people with the same DNA or similar STRs being in the same area? It's just not possible. But we take it a step further. We go to 13 STRs, and that yields a 1 in 900 trillion chance, which means it would never happen. No one would ever have. There's only been 110 billion Homo sapiens, 110 billion humans that have ever lived in the history of the world. And we're taking it to that 900 trillion level. So it's just not happening. They've tried it in court. They've tried to say, hey, you know what? It's possible for someone to somehow have the same exact DNA. It's just not going to happen. Okay. So they've actually created 13 STRs that they always look at, and these are the different 13 STRs. So there's one on the number 2 chromosome, there's one on the 3 chromosome, there's one on the 4, there's actually 2 on the number 5 chromosome, there's one on the 7, one on the 8, one on the 11, 12, 13, 16, 18, and 21 that we always look at from one person to the next. So using a thin glass column, the sample moves through the column filled with a liquid buffer and coated with gel polymer. And the electrical potential pushes the sample through and goes to a detector that's going to track the STRs that come out. And we have this capillary electrophoresis. So it's different than gel electrophoresis. We're using capillary electrophoresis, which is the new standard, which is going to reduce analysis time. It's going to be a lot faster. So commercial kits have made use of the amylogenin gene, which is different in each gender. The amylogenin gene is for tooth pulp, but it's used to determine the sex of the individual the sample came from. So even though it codes for this tooth pulp, it's different between males and females, and we can use that gene. We can look at that gene to tell, was this individual male or female. We also can do a YSTR marker, which is going to be used to decipher between 2 male samples. We're looking at the Y chromosome, and we're telling, all right, who's what. So if we have these samples and we want to know who did it come from, all right, we can look at the Y chromosome. We know it came from 2 different males. We can look at the Y chromosome and figure out which person did this sample come from. Mitochondrial DNA, you hear about mitochondrial DNA when they talk about familial relationships, because mitochondrial DNA always stays the same as it moves through a population from it's always the same as your mother. So everyone inherits the same mitochondrial DNA from their mother. So if you're male or female, you're going to have the same mitochondrial DNA as your mother. And if you're a male, your child is going to get your wife's or your significant other's mitochondrial DNA. And if you're a female, then your child is going to get your mitochondrial DNA that you got from your mother, that got from her mother, that got from her mother. So we can use this to look at lineage or familial relationships. CODIS is the software used to create the database of the DNA profiles, which is going to keep track of the 13 STRs of everyone that's on file. Contains profiles from DNA from convicted offenders, unsolved crime scenes. So we have DNA that doesn't necessarily have a name to it, and then any missing persons. And it uses, once again, the 13 STRs, which is going to yield that 1 in 900 trillion chance of someone else having the same 13 STRs. So practitioners only need about 18 cells to analyze a sample, so investigators should try to find any remnants of skin, which is our epithelial cells, blood, or saliva that might have DNA in it, might have some cells in it that we can use. For evidence collection, keep in mind we're still always taking photos. You're going to wear gloves for infectious disease control. Remember, if you're handling blood, you want to wear gloves. We're going to use single-use gloves, switch gloves from one piece of evidence to the next piece of evidence so there's no contamination. You're going to package everything separately in a paper bag or ventilated box so bacteria and fungi do not destroy the DNA. And you're going to collect any substrate controls. We also can use luminol, which is a chemical that is going to help investigators see blood that is hard to see or has been washed away. Luminol does not compromise STR analysis, so it's very useful in finding the blood and then using that blood for DNA evidence. So keep these things